Good morning everyone. Today I will be discussing about development of the nervous system. The development of nervous system will be dealt under the following headings. It is the neural tube formation, neural crest cells formation and the derivatives of it, development of the spinal cord, development of the brain, development of the ventricular system, and clinical aspects related to the development of nervous system. The competencies mentioned in the UG curriculum pertaining to today's topic are: number 1, describe the development of neural tube, spinal cord, medulla oblongata, pons, midbrain, cerebral hemisphere, and cerebellum. The other competency is about the congenital anomalies. They describe various types of neural tube defects with its embryological basis. Before studying the development of the nervous system, let us know what are the components of the nervous system. Under the nervous system, we have central nervous system and peripheral nervous system. Central nervous system comprises of the spinal cord, which is in the vertebral canal, and the brain, which is in the intracranium. Further parts of the brain are the forebrain, midbrain, and the hindbrain. The forebrain develops from prosencephalon, the parts of which are telencephalon and diencephalon. The midbrain develops from the mesencephalon. The hindbrain develops from the rhomencephalon, the parts of which are medencephalon and myelencephalon. Under the telencephalon, we have the cerebral cortex, basal ganglia, hippocampus, and amygdala. Under the diencephalon, we have thalamus and hypothalamus. The midbrain comprises of tectum and tegmentum. The hindbrain has metencephalon, under which are the pons and cerebellum. Under the myelencephalon, we have medulla oblongata. These are the parts of the central nervous system with the spinal cord included. Coming to the peripheral nervous system, we have somatic nervous system and autonomic nervous system. Under the autonomic system and autonomic nervous system, we have sympathetic and parasympathetic nervous systems. I will be dealing the development of nervous system in two classes. Today, in the first class, we will be dealing about formation of the neural tube called as neurulation and the clinical aspects or the congenital anomalies related to it, neural crest cells and its derivatives, what are open type of neural tube defects, development of the spinal cord, which includes histogenesis and functional organization of the spinal cord, the level of spinal cord at various stages of development in relation to the vertebral canal. Now let us see the formation of neural tube. A transverse section through the developing embryo shows the following three layers: the dorsal ectoderm, ventral endoderm, and the mesoderm. In the midline, we have the notochord already developed. In the region of the midline, the ectoderm develops a thick plate of cells called as the neural plate. The paraxial mesoderm growth on both the sides of the notochord elevates the ectoderm to form the neural folds. Between the two neural folds, there forms what is called as the neural groove. These neural folds further come close together and later fuse, leaving behind a tubular structure called as the neural tube. The cells of the neural folds get detached to form a mass called as the neural crest cells. This is a diagram showing the formation of the neural tube as discussed earlier. Now let us see the neural plate, which is formed from the ectoderm called as the neural ectoderm, gives rise to what all structures. gives rise to neural tube as we have studied earlier, and this forms the central nervous system comprising of the brain and spinal cord. The neural crest cells, which we have studied as the detached portion of the neural ectoderm from the neural tube, form the neural crest cells, which give rise to most of the peripheral nervous system. The other cells which remain in the ectoderm itself but are component of the neural ectoderm form ectodermal placodes, and they form the cranial sensory ganglia, hypophysis and the inner ear. The steps in the formation of neural tube are at about the 16th day of intrauterine life. The ectoderm dorsal to the notochord thickens in the midline forming neural plate. With the development of the paraxial mesoderm on either side of the notochord, the margins of the neural plate are elevated as neural folds. As a result, the center of the plate sinks down, creating a neural groove. The neural folds gradually move together towards the midline and finally fuse to form a cylindrical neural tube that loses its connection with the surface ectoderm. The process of this neural tube formation is called as neurulation. The fusion of the neural folds so as to form completely a neural tube starts in the middle of the developing embryo. It starts on the 20th day of intraembryonic life. Simultaneously, it proceeds cranially and caudally. The fusion at the cranial and caudal ends of the nerve is somewhat delayed, forming small openings called as anterior and posterior neuropores. These neuropores help the amniotic fluid to flow through the neural tube in the initial phase. The anterior neuropore closes on 25th day of intrauterine life, whereas the posterior neuropore closes on 27th day of the intrauterine life. By the time the neural tube is completely closed, it is divided into a cranial enlarged portion which forms the brain and a caudal tubular portion that forms the spinal cord. To summarize the formation of the neural tube, on 16th day of intrauterine life, there is formation of the thickened ectoderm in the midline called as the neural plate. By 20th day of the intrauterine life, the neural folds start to fuse in the middle of the developing embryo. By 25th day, the anterior neuropore closes. By 27th day, the posterior neuropore closes, thus forming a complete neural tube. During the formation of the neural tube, we have seen the cells at the tips of neural folds break away or get detached from the neuroectoderm to form the neural crest cells. Here you can see the neural crest cells, and these neural crest cells gives rise to the following structures. They are the dorsal root ganglia, sensory ganglia of the cranial nerves, autonomic ganglia, adrenal medulla, chromaffin tissue, melanocytes and squamous cells. You can see here they are giving rise to the melanocytes, cells of the dorsal root ganglia and ganglia of the cranial nerves. You can see cells of the autonomic ganglia, adrenal medulla, which is in the adrenal gland, the squamous cells which form the sheath around the axons of the nerves. We know that the neural tube formation fusion happens from the middle of the developing embryo. Later on it takes towards the cranial aspect and caudal aspect. The fusion of the neural folds cranially will help for the proper formation of the neural tube. If suppose there is a failure of the fusion of the neural folds in the cranial aspect, and there is a persistence of the anterior neuropore, then there is no formation of the cranial vault and the brain tissue is exposed externally. The condition is called as anencephaly. It is because of the failure of the cephalic part of the neural tube to close and associated defective development of vault of the skull. There is no bone covering this, which is called as the vault, producing anencephaly. There is one other condition called as rachis, a severe form of spina bifida. This is happening in the lumbosacral region. Anencephaly is happening in the cranial region, but rachis is happening in the lumbosacral region. This is the region where there is a posterior neuropore region. So failure of that posterior neuropore completely getting obliterated and the dorsal vertebral arch not fusing together leads to rachis or a severe form of spina bifida. In this slide you can see this is the anterior end of the growing embryo or cephalic or the cranial end. This is the caudal or the posterior end of the developing embryo. Here will be the anterior neuropore. This is the site of posterior neuropore. These are the somoids on either side of the developing neural tube. The fusion of the neural fold starts from the middle here, then it extends cranially as well as caudally. The failure of the fusion of the neural folds leads to the following anomalies. They can be open type of neural tube defects, which are anencephaly. You can just observe the area which is defective here leading to anencephaly. This marking is important, just observe that. And if suppose craniorachisis is supposed to happen, which means to say the cranial region is involved as well as the rest of the portion of the vertebral column is also involved, where the neural tube is exposed externally, then the condition is called as cranio, refers to involving the cranium, and rachis cases involving the rest of the portion also because of the split of the dorsal vertebral arch and the brain tissue getting exposed externally. But if suppose it happens only in the caudal aspect, that is the lumbosacral region, it is called as rachis cases or severe form of open spina bifida. These three varieties are the neural tube defects which are open type. But you can also see some closed type of neural tube defects wherein the brain tissue is not completely exposed to the external aspect, but there is a herniation of the brain tissue which is covered by the skin here. We have seen in the earlier slide the reason of why anencephaly and rachis congenital anomalies happen. But let us see the features of anencephaly. They are the vault of the skull is absent, that is the bony covering of the cranium is absent. The brain is represented by a mass of degenerated tissue and it is exposed to the surface. The cord is open in the cervical region. Cord refers to the spinal cord is open in the cervical region. The appearance of the child is characteristic with prominent eyes bulging forward and chin continues with the chest as there is no neck formation. These are the features what you can see in an anencephalic baby. The features in a rachis cases or a severe form of spina bifida baby, there is failure of dorsal portions of the vertebral arches to fuse with each other. It is in the lumbosacral region that this anomaly happens. The neural tissue is exposed to the surface. Occasionally, the neural tissue also shows considerable overgrowth in this region and it may become necrotic before or after the birth. Now, let us study the development of spinal cord. As we all know, the neural tube has a cranial dilated end, you can see here, which gives rise to the brain, and a caudal tubular structure that gives rise to the spinal cord. When a transverse section of a spinal cord is taken, we can see the following. This is the posterior aspect or the dorsal. Here is the anterior aspect. These are the lateral aspect of the spinal cord. Dorsally we can see here the dorsal root along with its dorsal ganglion. This is mainly sensory in function. Ventrally you can see a ventral root which is motor in function. Both join together laterally to form the spinal nerve. In the main tissue of the spinal cord you can observe centrally it has a H-shaped structure which is nothing but the gray matter, which is neuronal cell bodies. The periphery of it is white in color, which is having the myelinated axons. Now, histogenesis of the neural tube, which refers to the formation of various cells and axons within the developing neural tube. The neural tube mainly has three zones of development from internal to externally. They are the matrix zone, innermost, the middle mantle zone, and the outermost marginal zone. Let us see why the regions are named so, or the zones are named so, and what are present in each zone and how it develops. According to the current theory, the wall of recently closed neural tube consists only of one type of cell called as pluripotent neuroepithelial cells. This, what we call it as the matrix zone, has only one type of cell called as the pluripotent neuroepithelial cells in the initial phase, but it constantly divides itself to give rise to a pseudo-stratified neuroepithelium, which surrounds this zone. The zonal appearance of the matrix or mantle and marginal zone is because of the proliferative cycle that is happening of the pluripotent neuroepithelial cells, and this sequence being termed as interkinetic migration. The cells migrating from the inner aspect of the neural tube towards the surface of the neural tube, which is called as interkinetic migration. The pluripotent neuroepithelial cells, which are the innermost cells, give rise to the neuroblast cells that settle in the mantle zone, which forms the future gray matter of the spinal cord region. The axons of these cells traverse through the next zone called as the marginal zone, which forms the white matter as it contains the axons which are myelinated. The development of functional columns of the spinal cord. The functional columns refers to whether they form sensory or motor components and whether they supply the somatic or the visceral structures in the human body. Let us see here. The cross section of the developing spinal cord has a vertical slit-like central canal. This is the dorsal aspect. There is the ventral aspect of the developing spinal cord. A sulcus limitans develops in the middle, which separate the dorsal portion of the spinal cord from the ventral portion. The dorsal portion of the spinal cord we call it as the alar lamina. The ventral portion we call it as the basal lamina. Now coming to the alar lamina, which is mainly a sensory component portion, which has two columns. They are the first one here, which is General somatic afferent column. Afferent column refers to it carries the sensory information from the periphery to the spinal cord. The other column, what you can see, is called as general visceral afferent column. So it receives the sensory information from the viscera of the body. Coming to the basal lamina portion of the spinal cord, we can see two such columns here also, but they are mainly concerned with the motor function. The name is accordingly. We have the general somatic efferent column, and general visceral efferent column. By this you can understand that the dorsal root ganglia and the alar lamina of the spinal cord, which has the two columns, is mainly concerned with sensory input. The ventral portion of the spinal cord region, which we call it as the basal lamina, that is anterior to the sulcus limitantium, is mainly concerned with motor function. So the ventral root here, you can see carrying the motor fibers to the periphery. Now, having studied the different functional columns that form in the spinal cord, let us see the function and where they are located. The cells of the alar and basal lamina are arranged into two longitudinal columns. These are the columns of the alar lamina, these are the columns of the basal lamina. Now, the two afferent columns referring to the afferent refers to those present in the alar lamina. They receive axons from the dorsal root ganglion. And what do they do? Let us see. The general somatic afferent column. You can see here the yellow in color, the general somatic afferent column. It extends throughout the spinal cord, right from the cervical till the coccygeal, and receives impulses from superficial and deep receptors. That is mainly somatic in origin. These sensations are somatic in origin, maybe from the skin or from the muscles. Now let us see the second column. It is general visceral afferent column, green in color. It is confined only to the thoracolumbar and sacral regions, and it receives impulses from the viscera as well as the blood vessels. It is mainly concerned with the autonomic nervous system. The first one is concerned in bringing the sensory impulses from the somatic nervous system, that is a musculoskeletal system. This will be concerned in bringing impulses from the autonomic nervous system. Now let us see the efferent columns, which are here colored in blue and chocolate in color. The two efferent columns of basal lamina, they give rise to the motor fibers. These are the motor fibers which pass in the ventral root. The first column here we can discuss about is general visceral efferent column. It is general visceral efferent column, which is in blue color. It is present in the thoracolumbar and sacral regions. The same as we have seen earlier. So this is concerned mainly with providing the motor fibers to the viscera, glands and the blood vessels. The general visceral efferent column refers to autonomic motor nervous system. It refers to mainly the autonomic motor nervous system. Coming to the second column called as general somatic efferent column. You can see here in chocolate color, it is general somatic efferent column. It extends throughout the spinal cord and provides fibers to the skeletal muscles. Okay, somatic motor nerve fibers. These are the cell bodies of which are in the general somatic efferent column. So the ventral root carries the motor fibers, maybe the autonomic motor fibers or the somatic motor fibers. The position or level of the tip of the spinal cord is of importance because while giving spinal anesthesia in lumbar region, we have to know at what level the spinal cord ends. Now let us see here. During the 8th week of intrauterine life, the level of the spinal cord, that is the tip, is in accordance with the length of the vertebral column. That is, the spinal cord extends till the coccygeal region. By 24th week of intrauterine life, the spinal cord recedes and it is at the level of S1 vertebral level. And at birth, the tip of the spinal cord is at the level of L3 vertebral level. This shift in the level of the spinal cord towards the cranial aspect is because of differential growth of the vertebral column with respect to the spinal cord, which means to say the vertebral column grows at a faster rate than the spinal cord. Here, from birth till the adult, the spinal cord further recedes and settles down in the adult at the level of L1 vertebra. Now, if suppose you have a child to be operated and an adult to be operated, and you opt for a lumbar anesthesia, you have to be cautious in children with respect to the level of the spinal cord, because the level at which we give spinal anesthesia is in between L3 and L4 vertebra or L4 and L5. If the spinal anesthesia is given in the newborn at this level, it may damage the spinal cord. So knowing of the positional changes of the spinal cord with respect to the age is of clinical importance. Thank you everyone.